Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we are looking at polysaccharides. Polysaccharides are high molecular weight compounds made of monosaccharides and the derivatives joined together by glycosidic linkages, the same glycosidic linkages that we looked at in the section or previous scribblecast on disaccharides. Homopolysaccharides are those polysaccharides made up of a single kind of monosaccharide, while heteropolysaccharides are those polysaccharides made up of two or more different constituents. Most carbohydrates found in nature occur as high molecular weight polysaccharides. Starch, glycogen, cellulose are polysaccharides, polymers of the monosaccharide D-glucopyranose. D-glucose is the most abundant carbohydrate building block in the biosphere. Polysaccharides have three primary functions involved in structural roles, storage roles, or specific functionality roles. The first polysaccharide we're going to examine is a structural or architectural form. Let's get back to blue. What's called a structural form, sometimes also called a architectural form. And the first one we're going to look at is cellulose. Now, cellulose represents the most abundant naturally occurring compound on Earth. It accounts for 50% of all bound carbon on the face of the planet. It occurs throughout the plant kingdom as a component of cell wall, and frequently it is the major component of that cell wall. Cellulose is a linear unbranched polymer of beta-D-glucopyranose. Beta-D-glucopyranose. Beta-D-glucopyranose units are joined through what is referred to, if you recall from disaccharide chemistry, a beta 1,4 linkage. So, let us draw. Here is our D-glucose. And we're going to have a repeating beta 1,4 linkage. And it keeps repeating. Okay. Now, these polysaccharide molecules are organized into bundles of parallel chains, which are cross-linked by hydrogen bonding. These fibrils are twisted and insoluble in water. When cellulose is reacted with iodine, there's no color change. Humans, interestingly, do not have enzymes that can hydrolyze the beta 1,4 linkage. As a result, Humans cannot consume grasses and other forms that rely on beta-D-glucopyranose for food. The cow, for example, is a ruminant, can digest cellulose since bacteria present in the rumen, one part of the stomach, form cellulases. Various insects, snails, fungi, algae, and bacteria also produce cellulases. Cellulose nitrate has been used in the making of celluloid, photographic film, lacquers, etc. Cellulose acetate has been used in the making of photographic film, acetate, and rayon. Another example of a structural form is chitin. Chitin is a linear polymer of beta-D glucopyranose in which the OH of the second carbon, I will circle it, has been replaced with an N-acetyl group. Therefore, chitin is a linear polymer of N-acetyl D-glucosamine. N-acetyl D-glucosamine. Each of those units, which are found in cellulose, have the OH of carbon 2 replaced with this N-acetyl group. Everything else remains the same. So I'll just draw one fragment of the repeating unit for chitin. So chitin is just like cellulose in that it has a beta-1,4 linkage. It's made up of beta-D glucopyranose units in which the second hydroxyl has been replaced with this N-acetyl group. Chitin makes up the shell of crustaceans and scale of insects. Each year, approximately 10 billion tons of chitin is produced. Chitosan, which is derived from chitin, is a tough water-absorbent film which is biocompatible. A biocompatible material that can be used in the treatment of burns. We can also mold contact lenses from it. Coating seeds and plants with chitosan has shown increased disease resistance and thereby increasing crop yields by some 10 to 34%. Chitin and cellulose represent the two major structural forms that we find for polysaccharides. In terms of storage and nutritional forms, or nutritional forms, the first form that we're going to look at, or the only form we're going to really look at, is starch. Starch is the second most abundant polysaccharide in the biosphere. It's found in the granules of cytoplasmic cells, and when there is a surplus of glucose, enzymes link the glucose to the ends of starch chains. And when there's a deficiency, they release it. Starch exists in two forms: 20% amylose, which is hot water soluble, and 80% for form that is referred to as Amylopectin. Amylopectin is insoluble in hot water. Insoluble in hot water. So let's begin looking at A, amylose. Well, amylose is similar to cellulose in that amylose is a linear molecule, but instead of beta D-glucopyranose, we use alpha D-glucopyranose. So it's a linear molecule of alpha D-glucopyranose. Repeating. It consists of alpha 1,4 linkages. So we have the alpha 1,4 linkage. And it repeats. Now if I draw amylose, and I'll just draw 3 repeating units, now we can finish off our structure. Okay. It has molecular weight from 5,000 grams to 500,000 grams per mole, so it's a very long repeating chain with this alpha 1,4 linkage. The structure, though, is a helicocoiled conformation, and in water, amylose forms hydrated micelles. When reacted with starch, you get a very deep blue color. The blue color results from electronic interactions with the I2 molecule that inserts into the helical coil. For example, if you had this as a helical coil and you had 2 iodines here, let's see if I can make it a little larger. And so on. The iodine actually inserts into the helix. Now you know why this is called a scribble cast. And electron transfer reactions of the iodine that inserts into helix. Gotta be able to draw this better. I may give up after this one. Try it maybe the other way. Yeah, I'm giving up. So much for my artistic ability. The I2 inserts in the helical coil that is formed by the linear chain of alpha D glucopyranoses connected through this alpha 1,4 linkage. Now, most plants and most plants contain 2 enzymes called alpha and beta amylase. These are enzymes that will break, the alpha 1,4 linkage. Now, first thing I have to make clear is that we did a lot of discussion about enzymes and cleavage of disaccharides in the last scribblecast. There we used terms alpha and beta to designate the type of linkage they cleave. The alpha and beta here have no such designation. Alpha and beta here simply distinguish between the enzymes. It's like saying enzyme 1 and enzyme 2, or A and B. The alpha beta here has nothing to do with the linkage they cleave. Amylase cleaves alpha 1,4 linkages. Alpha amylase cleaves the linkage and generates a mixture of glucose and maltose. So alpha amylase gives you a mixture of glucose plus maltose. Beta amylase cleaves alpha 1,4 linkages, but it gives maltose only. So these two enzymes will cleave alpha 1,4 linkages. They will not cleave alpha 1,6 linkages. Okay? They cannot cleave that. Starch is digestible by man due to the presence of salivary amylase and pancreatic amylase in digestive secretions. And starch gets completely degraded to glucose. Now, what about amylopectin? Well, let's just erase this and leave our structure of amylose up. We'll use it. Okay. You might wonder, why did I mention alpha 1,6 linkages? We've never seen alpha 1,6 linkages. And that's true. We never have. Okay. But amylopectin differs from amylose only in the fact that every 25 to 30 residues, 25 to 30 residues, you get a branch occurring through an alpha 1,6 linkage. Ah, there it is. In other words, as we go down this lane and the chain and we hit carbon 25 or glucose unit 25 or 30, what happens is we'll get a new branch occurring. And this starts a whole new chain. So every 25 to 30 units is an alpha 1,6 linkage that produces a branch. Now, reaction of amylopectin with iodine yields a red-violet color. Alpha and beta amylases, as stated, can't cleave the alpha 1,6 linkage. So hydrolysis of amylopectin with these enzymes yields highly branched polysaccharides with intermediate links. They're called dextrins. So if I use amylase on amylopectin, I get dextrins. I get dextrins. There are enzymes that will debranch and hydrolyze alpha 1,6 linkages in nature. Now, what's interesting about this structure is this structure of alpha 1,6 branching and alpha 1,4 linkages is also present in another storage or nutritional form, which is found in animals and bacteria. And that is glycogen. Glycogen has the exact same structure as amylopectin. That is, glycogen has alpha 1,4 linkages, and it has branching through alpha 1,6 linkages. The only difference with amylopectin is that these branches occur every 8 to 10 glucose units. And so glycogen is even more highly branched than amylopectin. Now, amylopectin is a storage form found in plants. Glycogen occurs in cells of animals and bacteria. It's primarily stored in liver, but also muscles. About 5% of the liver is composed of glycogen. Its structure is even more highly branched. So let's test my artistry again. If this was amylopectin, where you had some branching off the carbohydrate chains, glycogen would be far more branched. And so if you were to compare glycogen to amylopectin, you'd see something like this. Far more branching in glycogen than in amylopectin. Now, within the cell, the glycogen molecules degraded by glycogen phosphorylase into D-glucose 1-phosphate. Now, D-glucose 1-phosphate is an intermediate in a metabolic process such as degradation of glycogen to lactic acid in muscle glycolysis. And so glycogen is very important biologically. That leaves only the third functionality, and that is specific functionality. Carbohydrates often find themselves in conjunction with other molecular structures, such as fats or lipids, and they're referred to as glycolipids. We'll be talking more about glycolipids when we actually cover fats. What we want to discuss right now are glycoproteins. Glycoproteins are polysaccharide protein complexes. Glycoproteins can have anywhere from 1% to 80% carbohydrate content. Glycoproteins with 4% or more polysaccharide content are sometimes called mucoproteins. Now, glycoproteins are of key importance in cellular recognition. Very important in cellular recognition. Invading viruses recognize biological cells by the glycoproteins on the cell surface. Since viral attachment to a cell surface is the first step in infection, preventative strategy in the treatment of such infections would involve inhibition of such attachment. Understanding the precise nature of such interactions at the molecular level is key to the rational development of new drugs and vaccines that can be used as therapeutic treatment. So cellular recognition is achieved by carbohydrate units attached to proteins off cell walls. Glycoproteins also play a key role in the cellular recognition not of just viruses, but of antibodies. The glycoproteins of the antibody only fit the specific carbohydrate ends of the glycoproteins on the corresponding antigen. This is clearly observed in the compatibility of blood types. Now, I'm going to take a moment to try and hand draw some of the diagrams I've provided in class. And if I take a look at this, what do we see here? I have off of the protein O up. Okay, is that right? So that's an oxygen up, and then that one's down, that one's up. And okay, so that looks like, let's just try drawing it. I have a protein, so some protein here. And then off of this protein, it looks like I have a carbohydrate. And if I look at this, it looks like it is up. So I'm going to draw it this way, attached to the protein. That is up. This one's down. Okay, and that's carbon 2. That's hooked to another carbohydrate. Alrighty, so I have off of the second carbon, that's right, from the aromatic carbon, that is down. This one is up. And then that one is definitely up. And CH2OH is up, so it's a D sugar. What is this guy? From where he attaches, this one's up. This one's down. This one's down. And it looks like this is just a CH3 and it's down. So an interesting carbon fragment here. Now, what I want to draw your attention to is this unit. Off of the protein. When that is an OH, we have what is called type O blood. But if we replace that OH and we replace it with we attach that to it. This is down, up and up. And that's a CH2OH. So it looks like we replace the OH that's in the red box with what looks like an alpha D galactose unit. Then we get what is known as type B blood. Now, I don't expect you to draw these fragments. I'm only drawing them for interest. If we replace that OH that's in the red box with a galactose that is further modified, again, yes, it would be alpha. So it is a galactose, a D-galactose. But instead of carbon 2 of the OH, we have an N-acetyl group. If that's attached in place of the OH, we get what is known as type A blood. So type O blood has a carbohydrate unit consisting of a galactose off a protein with another carbohydrate fragment off of it. And if you replace the OH off the third carbon here, the one that's surrounded in red, a red box, with an alpha D galactose, you get type B blood. If you use an N-acetyl alpha D galactose, you get type A blood. Now, the cell surface glycoprotein difference of these red blood cells, right, will cause, in some blood types, will interact with the antibodies, and in some blood types, these antibodies will cause the blood types to clump together. And so these carbohydrate functions, these carbohydrates off the protein restrict which blood types may be provided by transfusion because of their interactions with the antibodies. It is these terminal carbohydrate units that distinguish the blood type groups, and by which the antibodies recognize the corresponding antigen. The antigen-antibody interactions are, of course, an area of intensive research with significant implications in all areas of biologically related sciences. So specific functionality, especially cellular recognition, is a very important aspect of carbohydrate polysaccharide chemistry. In the case of blood types, we're talking about a trisaccharide fragment. Okay. Well, that's it for polysaccharides, and that finishes off carbohydrates. I'm Dr. Brian Lloyd. This is my Scribblecast. I thank you for listening.